All right. Oh, welcome everybody. So if you haven't figured it out yet, we're very casual here. That's the whole point of this, is actually to really be more about creating dialogue than it is about pontificating about any particular topic. InnovationWell is part of Douglas Connect's outreach and about building community to foster collaboration and innovation. And we're doing these in a few different locations. Philadelphia, we did on this past week on Thursday, here in Cambridge, and we usually do a third one in North Carolina down in Research Triangle Park. And the topic that we're focusing on during this round is about data and precision medicine, and how do you think about problems about solving problems. And we're going to be hearing a few different perspectives on that. And what I thought I would do is one of the speakers that was meant to come today was unable to attend because of some business conflict at the very last minute. So what I thought I would do is maybe recap a couple of the points that came out from our Philadelphia meeting, and hopefully it'll kind of set your mind and some of the questions that were posed there and addressed, and hopefully we'll have some further dialogue around today. So this is the group that was at our Philadelphia meeting. We had Michael Liebman from IPQ Analytics. It's a company that he co-founded. He's also a professor of Drexel Medicine and several other places. We've Jacqueline Tironi from Childhood Cancer Data Lab, which is part of the Alex's Lemonade Stand Foundation. They're basically funding research into uncovering some of the underlying pieces of childhood cancers. And Lucas Boscheman and Gower Jamsed from Vertex Labs, they're located in Philadelphia. It's a small startup, but their focus is on data analytics, artificial intelligence, machine learning, and helping. It's interesting when I have a conversation with them. As much of the work that they do is analyzing data, it's helping organizations really think about their data, not only from what they have, but how do you generate data, and how do you use the data that you've got. So those that are involved in machine learning aren't just about trying to sell you the next best pair of shoes or sneakers. They're actually trying to solve some serious problems. So a few reflections on the Philadelphia meeting. So these were the topics or their titles, and I thought they actually captured it quite well. I loved Michael's talk title, which was Real World Evidence. Does it address real world questions? And I'll show you a few slides from his talk that kind of resonated with me on this. And that is, oftentimes when we are trying to study a problem, we'll gather data, we'll gather information, we'll run assays, and usually when we run those assays, we run assays, no big surprise, around things that we can measure. So I'll give you some of my perspectives. So my background is in pharmaceutical research, and we would study a whole variety of disease processes. And without exception, every pharmaceutical drug discovery program we would work on would have a series of biological assays. And those biological assays are meant to be surrogates for the disease process that we're ultimately studying. No big surprise there, right? And then if you sort of delve into why are you running this assay and this assay in a particular under a series of circumstances, and it reminds me of the old joke of, why are you looking for your keys underneath the light? Because they could be anywhere. It's, well, there's light over here. And the same seems to be the case when you're looking at surrogate assays. You assay the things that you know how to do, that you think are most closely relevant to the disease process. And so it kind of really, to me, that while that's in the discovery space, when we're talking about real disease processes, is the data that we're capturing truly relevant to the situation? And I'll give a couple examples that came out of that. The folks from Vertex Labs sort of posed the question: should we be revisiting the scientific method? And the scientific method usually starts with a scientist looking at a problem formulating a hypothesis, and then coming up with experiments that you could actually test that hypothesis. On the surface, that makes perfect sense. That's how I was trained. That's how you start uncovering some of the groundbreaking pieces. And one of the things that was posed throughout the talks on Thursday was, is it possible that if you have perhaps a domain expert too closely tied into the questions you're trying to ask, could that bias the outcomes that you're actually trying to come to? So, for example, if let's say I'm a nephrologist and I believe that kidney disease is just about high blood pressure and diabetes, and I don't think that there's any other, that's the 99 percentile reason why people develop kidney disease, and I'm designing experiments, whatever biases I bring into the situation are going to could potentially affect the outcomes. And so from their perspective, it was, yes, you want to have domain experts involved, absolutely, because you need to be certain that you're capturing important information that you know is relevant. But oftentimes there could be information that is not so obviously relevant. So, for example, as we all know, in many diseases, it's the rare situation that you don't have comorbidities. And as human beings, we like to simplify things down to, one or two things that we can look at a given time, and then try to draw some types of conclusions. The reality is that certain diseases that we look at, certain adverse outcomes that we look at, are hugely multidimensional, with elements that we don't entirely understand. So we perhaps need to rethink the kind of data that we capture, the quality of the data, and this is going to sound like a dirty word, metadata. So all the pieces. So if I'm a biologist and I'm talking to a medicinal chemist, for example, I run an assay and I give them an IC50. The chemist just wants the IC50. Well, as a data scientist, there's probably another 50 pieces of data that I want to know about that experiment, so that when 2 years from now we're on the same program and a new biologist was hired to run that assay and they get a different answer for that compound that they just ran, I can perhaps look and try to differentiate. So data for an immediate question may not necessarily be the data for the long term, if you will. And we should be thinking a little bit more broadly about who contributes to the individual pieces when we're designing experiments and collecting data. And Jacqueline talked about, can big data help us tackle rare diseases? Now, for those who are somewhat familiar with rare diseases, we're talking about patient populations that could be as small as 10 or hundreds that they have, maybe up to thousands. So as patient populations go, that's really small to draw large conclusions. And so the direction that they're looking at genomics information, which instead of having like hundreds of thousands of patients, they're generating much more detailed information about each of the patients and trying to look at differentiating elements. So it's a different way of trying to, I'll say, generate large amounts of data to tackle a very narrow field. One of the things that really stood out for me from her talk is, again, about, and a little bit of it comes back to revisiting the scientific method. The way we think about whether it's a disease or a particular problem is so what, here, let me rephrase. So one of the things that she brought out is that there's, I think if I remember the numbers right, that there's about 12 different cancers that they focus on. But underneath those 12 cancers, for the childhood cancers, there's actually about 100 variants of those cancers that they can striate those cancers into. And as a result, you'll have different treatment regimens for those different subtypes. And so if you call it, X, and in reality it's X 3.1, you could be giving a close appropriate treatment, but maybe not accurate enough. So how we call things, sort of that ontology of that we struggle with in almost every field, is something that when we think about data, we've got to take into account. And one of the things I walked away from that meeting on Thursday was the way we look at populations of information, or populations of data, I should say, and try to use that to characterize things so that we create, I'll call it the stereotypical patient, if you will, for a particular disease, can be very misleading. And if you look at deeper levels of the information that you may capture, you may see the striations that, like, as I'm referring to in the cancer case. And, very broadly, if you have enough technologies and enough data, do you actually get to solutions? And hopefully you kind of get the idea is that, no, the simple answer is, data and technology is not the answer. It's having the right technologies and the right data combined together. These are questions you've probably posed yourself for when you're embarking on a research problem. So are we really collecting the right data to answer the questions we are asking? I kind of refer back to the situation that we talked about earlier in the drug discovery world, where if I'm studying a disease and I'm simplifying it to a particular enzyme reaction, am I doing that reaction under the right conditions that actually simulate something that's downstream? Because the assay conditions may only work under conditions that are totally inappropriate for the disease state to be. So I may not actually be measuring the right thing, even though the biologists may tell me that I am. How much data is enough? Invariably, there will be data gaps, and how do you deal with those data gaps? And are we? This is, I think, a really important point, I would say, that we can think about now is, are we really designing experiments for now? Or are we designing experiments that will solve the problems that we have now, but also can be used in the future? So that introduced that concept of that metadata that I was talking about, is that it may not be enough to just give me, an IC50 value, for example. But if I have all this other piece of information, I can now start doing comparisons, maybe for a whole different disease state several years from now. And so I thought, Michael's been really gracious. He let me steal some of his slides because I thought he captured some points really well. And at the end of the day, so as we're going through today's discussions, keep those things in mind. And I really like this quote from Edward Deming: "If you do not know how to ask the right question, you discover nothing." And it's that classic situation of garbage in, garbage out. And so, posing, what do they say? A question well formulated gets you probably 85% of the way to the answer of the question you're after. And so, as we're thinking about the clinical outcomes we're looking for, trying to maybe approach precision health and precision medicine, we really need to be formulating the right kinds of questions, and that from there we can actually then start thinking about what is the right data that we should be gathering. I'm going to actually kind of go through these kind of quick. I came across this, so I'm a fan of Albert Einstein, as many people are, but I felt like this one was really appropriate, because too many times in science, what we try to do is we do try to simplify a problem down to its simplest cores, down to those 1 or 2 or 3 steps that we believe might be pivotal for having the outcomes that we're interested in. And that in essence is human nature. But for me, this is actually sort of like a yield sign, if you will, is that we should make things as simple as possible, but not simpler. Because if we reduce things down to just a couple variables, it can be very, very misleading. And I thought this was a really nice example from Michael. So don't worry about the details here. It's more about So this would be the example of I've got an assay that I'm going to run to evaluate a particular condition, and I break it down to a single assay, and based on the outcome of this, I'm going to say you've got a condition or not, or you're going to have an effect or not. But the reality is that this biological process that I'm trying to mimic is actually part of a much larger process, and without understanding how it interacts with this, I won't be able to necessarily understand, am I just dealing with one disease or possibly many? And then the reality is that our body is a much more complex collection of interacting biological pathways. So we sort of back to that last slide where it's, you need to make it as simple as possible, but if you make it too simple, to expect that this one interaction is going to be reflective of this larger system is probably naive on our part at this stage of the game. It also reminds me of that classic saying is that every model is wrong, but some are useful, right? And in drug discovery, that single assay probably got us 85% of the way to an answer that if we started going into looking at more complex systems, you can make headway, right? So you don't want to lose the forest through the trees, if you will, but I think it's about bearing in mind that the systems that we're trying to model and collect data on are really complex. And so I come a little bit back to that point earlier, is that if I have a domain expert and the domain expert, let's say in the clinical setting, says, I always measure X for this particular disease, if he's trying to design an experiment to study this, he or she is going to say, measure X. These other things are probably not so relevant. And we may need to pose the question. There's a lot of techniques out there now that allows us to address those kinds of things. The next couple slides, again, just reemphasize that same point. We can simplify the process of disease from, the risk assessments, and again, the specific details are less important as opposed to, you start with a risk assessment and you'll come up with a diagnosis based on the risk assessment, maybe a differential diagnosis. You stratify your disease, come up with a treatment options, apply it to a general patient population, and hope that your individual patient outcome will reflect what the general patient population will be. Now, I'm not a physician, so I want to make sure that caveat's out there. But having worked in the drug discovery space, this is how we oversimplify things. When the reality is that there's a lot of other interacting elements that play a role here. You've got the physician who is constantly looking at the patient and the treatment options and how patients are responding. You've got the pharma companies that are trying to provide solutions into this space. You've got, I don't want to sound too cynical here, but you've got payers and providers. Basically, the people, when you walk into a hospital, their objective is to get you as healthy as possible, as quickly as possible, and out the door as quickly as possible, in, I'll say, the most cost-effective way possible. And that's I'd love to believe that it's consistent with the way the physician would like to treat you, but from speaking with enough physicians, the two are not necessarily in alignment. And so you've got all these different contributing factors to the outcome. And when you start trying to do data analysis on let's say, patient outcomes based on treatment options, it's not just, I've got a patient with a series of characteristics and they took treatment X and they had outcome X. There's a lot of other variables that play in here that if we ignore, we may actually be missing the whole story. This kind of just elaborates the next layer down of items that you can think about in terms of information that could give you insights onto whether you had a successful outcome or not. And probably offline, if you'd like, I could give you a few anecdotal stories about from that I'm familiar with. And again, so for this slide, details less important as I would say the title, and that is the electronic health records that we capture really are not a gold standard. 503. Here I may be opposing a cynical view. Michael brought this out. I agree with it because I've got some experience in the space, having worked with a variety of physicians, and that is that it's quite interesting that in order for a physician to get paid, they need to work with the providers and the payers to make sure that they capture ICD-10 codes, basically that diagnose a patient for a particular disease. And if they come into the office and they present with certain symptoms, they put in a particular code, they run whatever tests are necessary, and the information that they capture will be reflective of the diagnosis, not necessarily all the details of what's in the what the patient is presenting with. And so while there's a lot of information there, I don't want to say that it's suspect, but it may not allow you to draw direct correlations or relationships between a specific disease at a particular state and time with outcomes that you're trying to measure. So now I'll put the caveat in there that your mileage will vary, right? So there are some hospitals and some settings that you're going to have exquisite detail. So for example, if you're in most clinical trials, you're typically going to have significantly more information captured and than you will if you go to your family physician and just go for a checkup because I'm having chest pains and a little bit of congestion in my chest, and have them capture information. So when we start doing data mining as data scientists, we've got to be aware of this, and the understanding that information, while it's oftentimes out there and available, needs to be looked at on a case-by-case basis. This is, I think I'm just reiterating the same point. I think everybody gets it, is that we need to be looking at asking the right question. We need to be looking at collecting the data, and not just for the question we've got today, because I may be thinking or formulating or framing the question in a particular way today, whereas it's going to be in about a month or a year from now, that question could change based on new information that comes out either from my office, my lab, or other researchers that I'm working with. The other piece, and maybe this comes back to that first slide where I showed the group that we brought together in Philadelphia, and that is bring diverse friends to the party. Let me check. So I don't think the problems going forward are just going to be solved by clinicians or by data scientists just looking at just pulling data, or by biologists or chemists, or it's going to be, you're going to need mixed teams. The days of one group thinking that they can pose the question, run the assays, and solve the problem. I think that ship has sailed, in my opinion, and only through significant collaboration and through significant interaction will you be able to really get to some of the higher level outcomes, the more complex outcomes that we need to get to. And one of the things that I would say some of our audience at Philadelphia was a little bit more on the data science side, and it was interesting to hear them when I was talking with them, the frustration that they were having with the data that they had access to, and here's a common statement: if only I could have had a conversation with the researcher when they were designing the experiment, I could have helped them figure out how to create more complete data sets, or how to capture the data, or the kind of data that would be useful for downstream analysis. And so that's just a common 60, yeah. Just a common frustration. And so sort of back, maybe a little bit back to the beginning. Again, for every complex problem, there's an answer that's clear, simple, and usually wrong. So simple is good. Too simple is we're probably going in the wrong direction.